grade 12 Mindsetters. I am back. It is 2:30 and we're doing life sciences. I am Lune and joining me is Cheryl. How
Hello,
are you,
Lune.
Cheryl?
I was surprised, that's fine.
And I know I'm surprised, but they don't. So Mindsetters, what we're doing today is that we're doing life sciences. Hey, Cheryl.
Yes, we're doing
what
life
topic
sciences.
are we
We're doing plant and animal responses.
All right. So Mindsetters, you know that we have a competition going on. It's been running for our whole spring school. So basically what you need to do is stop the clock. So how you do it is you tag yourself or share the post. I will post it on our Facebook page, guys, so don't sweat. It's on our Facebook page. All you need to do is tag yourself or share the page and with your lucky number from 0 to 9. And what we'll do is we'll check now. Our clock is on 6. We check the last digit of our Facebook likes. So Cheryl, if you could just point to 6. No, just
there.
Show them there. You see Mindsetters, it's 6. So all you need to do is keep on asking your friends to like if your favorite number is not 6 until you get to your favorite number. Tag yourself on the post that I will post on Facebook and we'll see if you're a winner. We will announce the winner on our next show. So we have an overall winner. So the first show we had 1. That's where it stopped. You will be sent to a lucky draw. Today's this show is a person who's all the people from this show who have been sent to the lucky draw will be sent to the final lucky draw, and then at the end of the show you must watch at 4 o'clock, guys. We might announce you as a winner of that very special Learn Extra hamper. So with all that said, thank you so much for tuning in, and I'll hand it over to Cheryl.
Thank you, everybody. Please remember, go to the Facebook page, go liked, and you could be a winner there. Okay, carrying on to the more serious stuff. Afternoon grade 12s, welcome to the Spring School edition. I hope you learn a lot this section. We are looking at plant and animal responses. We're going to be going through a past paper from February and March. Please remember, past papers are an excellent tool. I went on to the Department of Education last night, also just to have a look at a few of them. They are there available for you. The memos are also there. It's a wonderful tool. And I see the Department of Education, I've also got a clock counting down how many hours actually it is until your final exam. So either you're going to get a big fright and wake up, or you're just going to be chilled and really think maybe it's a bit of a long time. But let's get started. All right, we're going to start off with a few of the multiple choice questions. Your teacher might have given you lots of hints or whatever how to approach the exam. I'm also going to give you a few of my hints. It's a case of take it or leave it. If you feel more comfortable doing it in your own way, please. All right, just you are more than welcome. Or if your teacher's given you a specific way in which she wants you to be able to answer the paper, all right, I'm just going to start off with a few hints as we go through, all right, some of the questions from the February and March of this year, all right, the supplementary exams. When you get your paper, you should get reading time, all right? And some people, they rush to the back and they start at the back, but I always think the most important part of the paper is the section A, your very first section. Reading through your multiple choice, reading through your terminology, because when you start reading through the multiple choice, the answers are there already for you. So it's almost like when you're reading, things are going to start snapping. You're going to see words, and then your brain's going to start firing, and you're going to start to, because quite often some of the key words in the multiple choice and terminology can actually help you later on with what they call the longer questions. Okay, so I always find it's much easier, read from question 1 through to question 5. And only if you see that maybe time is short that you jump questions. It's sometimes much easier if you do it in the pattern. And remember, question 1 is your easier section. All right, the multiple choice, the right answer is there. If you are not sure, you guess. You never leave out a multiple choice question. All right, you always, if you don't know what it is, you just guess it. No minus marks. If your answer is wrong, it's wrong. Okay, let's start off with a few from the paper from the March. All right, and we're going to get started. Right, please remember, it's A, B, C, or D. Right? Do not write the answer out. If you write the answer out, the markers are not going to mark it. Okay? Because the instructions to you are very clear. You have got 4 choices, and you must use a letter. Look here. All right? If it says, choose the correct answer and write only the letter. So let me get my pens going over here. All right, pen color. Oh, here we go. All right, please, very important. Write only the letter. Don't write the whole thing out. Okay, you are wasting time, and then it shows us that you actually don't understand your work. If we start with the question, the first question, number 1, right, drinking a large volume of water will lead to. Now, when this already should start to tinkle the brain from last year's excretion, right, when you did the kidney. Okay, and here we're going to talk about ADH. This year, we're looking at the endocrine system, the hormones, all right? And last year, it should have triggered a bit of a, oh, I remember that from last year, all right? Those of you who were paying attention, that it was the kidney. Okay, now, our question is, drinking a large volume of water will lead to. Now, remember, let's go back. When we talk about ADH, I'm going to write over here, antidiuretic hormone, okay? What do we need to know? ADH will work when we are thirsty, all right? ADH will work when we are thirsty. That means that the body doesn't have enough water, and ADH makes the tubules more permeable, all right? So, look here, drinking a large volume of water. All right, we have lots of water. If we're drinking a lot, does our body need to take a lot in? No. All right, so the first thing we're going to look at, I do not need ADH. So the first option, let's look for a decreased. All right, I see 2. I see B and I see D. All right, now I could get a little bit confused here, but let's carry on. Decreased production of the ADH and kidney tubules become less permeable. Yes, all right, I don't need to absorb the water. I can let it go. I can make permeable means I shut it off, all right? I shut it off. Permeable means allowing it to go through. So what is my answer going to be? Definitely number B, because number D, more permeable. No, I'm drinking enough water, right? I've got enough. I don't need my body to absorb. So I don't need ADH, and I don't need to make my kidneys less, all right? There we go. Next one. Sensory data, cerebellum. All right, when we think of cerebellum, I always remember here the parallel lines, and to be able to walk on it, all right, what do you need? Balance and coordination. All right, that's how I always remember the cerebellum. So it says sensory data supplied to the cerebellum. So in other words, any impulse supplied to the cerebellum is used to initiate, that means start. All right, and why is it wrong? Because the cerebellum does not initiate voluntary movement, but the cerebrum. Coordinates motor movement. What did I say I needed to walk on those 2 lines? I need coordination. So I've got a tick, but let's carry on reading. Provide conscious information on muscular movement. No, where does my conscious information? My cerebrum. And lastly, number D, control reflexes. No, where's my reflexes? In my medulla oblongata. All right? Knowing the functions of the brain. So as I said to you, for those, lots of you get confused between cerebrum and cerebellum. So if you remember parallel lines, I need to balance to be able to walk on those lines. And what do I need for balance? Coordination. Okay, next one. When you see these, all right, warning lights are going on, plant responses to the environment. So we're talking about plant hormones, auxins, and all of those things. Let's see what our multiple choice question. Okay, now remember, it's telling you already that question 1.3 and 1.4 refer to this diagram. Okay, so what they're showing you over here, all right, is a tip of a shoot, and they've obviously cut the top off, and they want to know which growth hormone is most abundant. Which hormone do we find at the top of growing, all right, of the tips of roots? And we're going automatically, auxin. All right, auxins are responsible for growth upwards, okay? growth upwards. The rest, the gibberellins, the abscisic acid, and the cytokinins, also for growth, but usually in the ripening of fruit, producing a flower, all right, and growing of shoots. No, we're talking about at the apical meristem, at the top. All right, now the next question, all right, talks about phototropism. Remember, light, all right, phototropism, the movement of a plant in response to light. Now, if we have a look here, all right, this plant, all right, is being, look here, light from one side. So light has only come from one side. If we've got light from all areas, then my hormone stays in the middle, okay? But as I always say, all right, what does auxins is like Darth Vader, all right? Auxins like the dark, all right? I think of Star Wars. I don't know what you guys think of. It's the, I don't know, what's the Superman, Spider-Man, whatever, one of their enemies, okay? All right, so here's the light. Where do they go? They go to the dark, all right? They are photophobic. They're scared of the light. They move away. All right, so our question is, after the light exposure from one side for several hours, where will the highest percentage of growth hormone be? 1, 2, 3, or 4. Where did I say it wants to go? To the side of the dark side. As I always call it, Darth Vader. All right, and that will be number 3. Remember, auxins, where do they want to go? They want to go to the dark. They're afraid of the light. And once they go across to the dark, all right, remember, now they're all on this side. Let me just get rid of that. Now they're all on this side, so where does growth occur? There, because they can't grow, all right, uniformly. All of them are here. Look there, look at that. Look how much growth that is. And look how much growth that is, because they've all gone to the side, the dark side. Okay, they sound like evil roots. Okay, reflex action, all right? Reflex action. As soon as you see the word reflex action, you need to be thinking of your 5 points. What are your 5 points? Okay, number 1, stimulus. Okay, and then where does the stimulus go? Sensory neuron. Me and my sensory neuron. Where does a sensory neuron go? An interneuron or a connector? It's fine. Then where do we go? We go to a motor. And where do we go? Effector. Right, a reflex action, remember, is your quick instantaneous reflex, usually something painful. And you don't want it to go to the brain. It will take too long. So your question is, the following events occur during a reflex action. Which is the correct sequence? They've given you the 4, all right? Here, 1, 2, 3, 4. Now they want to know the sequence. Let's put it in over here. So I started off with a stimulus, all right? A sensory organ is stimulated. That is number 1. Where does that impulse go? Along a sensory neuron. Okay, because we need to sense it first. Then what happens? It must come back, a motor neuron. And what do we need to do? We lift up our hand or our foot. So that is number 4. Okay, so let's go back to our answers. We are looking for something that starts with a 2. All right, so we're ready, we know. C is out and D is out. All right, after number 2 is number 3. After number 3, all right, number 4. So there we go, that's it, voila. Okay, so what are we looking at? 2, 3, 4, 1. 2, 3, 4, 1. There we go. Number A is the answer. Then the last one, all right, in this question again, goes about regulation of body temperature. If we have a look at the flow diagram, all right, when there is a decrease, all right, in the human body temperature, so we are cold, all right, we are cold. which one of the following pathways shows the correct response? All right, by blood vessels in the skin. When you are warm, when you are cold, opposite things happen. Okay, so decrease in body temperature. There we go. What do we want? Dilation means bigger, constriction means smaller. We want constriction. The smaller they are, the further away they move from the top of your skin. Remember, how do we lose heat? All right, we lose heat through convection, conduction, all right, radiation. So we don't want to lose heat. So what do? We make our blood vessels very, very small. They move away from the surface of the skin. Aha, and what happens? All right, decreased blood flow to the skin. We don't want to look all moist and pretty and shiny when we're cold, all right? This is where we need our heat. So we take it away from the surface and we direct it into, all right, the most important body. Are we ready for a break?
A break. Okay, yeah, we can take
I see
one.
the clock there, 0, 0,
All right,
0, 0.
Mindsetters, we are going to take a break. Just a quick shout out to Masilo and Kennedy and Calvin. Thank you so much, guys, for tuning in. And a special shout out to Callan. Callan, Cheryl's daughter. Hi, Callan. We see you and you see us. Thank you for watching and thank you for being here. Mindsetters, let's take a quick break and we'll see you straight after this. Welcome back, Mindsetters, from the very short break. I hope you guys are nice and refreshed and you're still on par with us. Remember to hit us up on Facebook, facebook.com/LearnExtra, or Twitter @LearnExtra. Ask us any questions you may have, and remember to enter that competition because you could stand a chance to win a very special Learn Extra hamper. We do have a question, actually, Cheryl.
Do we?
From Shikash. Yes. Shikash is asking, could you please explain geo and phototropism?
I can. All right.
Is that how you pronounce
I can. Yes.
Yes,
All right.
that's the only question.
Is that the only question?
Yes.
I'm going to go back to the...
Okay.
What's her name? Sorry.
Shikash.
Shikashi. Oh, let me go back quickly. Hang on. There we go. There. Okay, Shikashi. All right. Let's find a nice little blank space, and then we can... Let me go back to this question. All right. Okay, Shikashi, let's go about phototropism and geotropism. All right, phototropism is the movement of a plant, right, in response to light. So what is positively phototropic is our stems, okay? What do we want to do? We want our stems to go up, and we want our roots to go down, okay? So stems go up in response to light, okay? And light is, we always use the word photo, right? So photo, meaning light, tropism, meaning movement. So our stems must go towards the light. Remember why? We want photosynthesis. But we don't want our roots to grow upwards, right? Otherwise, we wouldn't be able to plant into the ground. Where do they get their nutrients? Where do they get their nourishment? From the soil, okay? So we want roots to anchor. They're going to go downwards, okay? Downwards out of the light. So they are negatively phototropic, and they go down. What goes down? Gravity pulls them down. All right? And in doing so, all right, that is why they move down. Now, the light, we have auxins at the growing tip that move upwards towards the light, and our roots the same. We have auxins at the growing tip, which are stimulated by gravity. That is why geotropism, gravity, all right, roots will go down, and my stems, phototropic, will go up. So I've got stems going up so the leaves can photosynthesize, and I've got roots that are going down to anchor the plant and to absorb water. I hope I've answered your question. All right, we're going on to the next short question. All right, question number 2. Oh, there we go, me and this board. Okay, it is a question on insulin. All right, I think, guys, when it comes to the hormones, there are certain more popular questions than other ones. The insulin one is one, and the other one is the adrenaline. All right. So you might have one of them in the multiple choice and a longer question, but you must make sure that you know your insulin, all right, as well as your adrenaline. We're going to now focus on one of the questions related to insulin. Okay, let's have a look at the graph here. Okay, the question is, the blood glucose level is regulated by hormones. All right, oh, let me go pink, I like pink. All right, hormones. So there already it's telling you it's plural. Okay, and if I were you, I would be making notes and I would be saying, what are the two? It's insulin, all right, and glucagon. All right, when it comes to this diagram, it's a lot of glues. All right, there's glucose, there's glycogen, there's glucagon, so it can be very, very confusing. So pay particular attention, all right, to which is which. Okay, so let's start off. Glucagon, all right, is the hormone that is going to convert glycogen to glucose. Okay, gon is the glycogen, and then it gets going to glucose. So let's have a look. The human body attempts to maintain a glucose concentration of approximately 1 milligram per milliliter of blood. Okay, the graph below shows fluctuations. It can be quite a big word, all right, it means changes. Okay, fluctuations, when we talk about that, all right, that is a fluctuation. So we're looking at a human's blood sugar level over a 5-hour period. Okay, let's have a look over here. If we have a look at the graph, there's our 5-hour period, all right? And if we look, so this is my independent variable, and this is my dependent. All right, time was given, but the sugar level, that can change. Okay, so if you have a look over here, what you're looking at is that the sugar level changes over time, and we know that, all right? Think about it. Again, see how very often some of the work you did in grade 11, right, is relevant to the matric. So you need to pay attention in grade 11 as well. Tell all your buddies who are in grade 11, they must make sure they know, because it's gonna come back to bite them in the bum later. Okay? So what do we have? All right, we have what changes your insulin level? When you eat. All right? When you eat. So when you eat something, the insulin levels go high. And remember, too high levels or too low levels is dangerous. But when it goes too high, what do we need to do? We need to change that glucose, all right? And we need to store it as glycogen. But what will happen? What is that glycogen? It's stored energy. Maybe we need to dip into that storage. So now we need to get it back to glucose again, all right? That is going to be glucagon. So when we look at our graph, all right, if my glucose levels go up, right, I've probably eaten something, and when they go down, what's happened? We have converted glucose to glycogen. So let's see over here. From the information above and your knowledge of hormones, name the hormone released at B and D. Okay, have a look here. Blood glucose was high, and then at B, it went down. What makes the glucose level go down? Insulin. Okay, because what happens? All right, glucose, you don't have to write this, but I'm just trying to explain, gets stored as glycogen. All right, then at D, what happens over here? Then our levels go down again. All right, so what happens at D? We now, all right, they're going to start to go back up again. So what do we see at D? The level's starting to rise. What is insulin's opposite or antagonistic? Glucagon. Okay, look at all the glyphs. Now it's the opposite to that. It takes your stored glycogen, all right, and it converts it to glucose. Okay, there we go. Insulin, glucagon. It can only be one insulin or glucagon. All right, so you need to remember, when my levels are high, insulin brings my levels down, glucose brings my levels up of glucose. Okay, remember, insulin, glucose levels must go down. Glucagon, my glucose levels must go up. Okay, next question. Aha. Right, you can see I was preparing you for the next question when we looked at the graph. It says, now look at the first thing. Okay, the individual ate a meal at point D. Okay, look at the sugar levels. They were nice and low. Okay, explain the pattern of the graph for the next 2 hours. Explain. In other words, tell me what happened. All right, let's go and have a look at the graph. There we go. Right, number D. Okay, after it ate number D, what happened? I'm going to write it down, but let's just have a look. It increased, not it, okay, let me be more specific. The glucose, because there they are, tells me there, the glucose level increased. Okay, for how long? Let's work down here. Tell me, I want to know the time period. Okay, so the glucose level increased after the first hour of eating. So we write that. That's our first observation. We're explaining. The glucose level increased, rose, all right? After, because this question is actually, all right, for 3 or 4 marks. Okay, so you can't just say it rose. Glucose level increased after eating, okay, for 1 hour. Okay, give me the reason why. Insulin converted, all right, okay, for an hour. Okay, then let's say it drops, all right, for the second hour, and the reason here, insulin converted the glucose to glycogen. All right, explain the pattern on the graph. Let's go back again. First hour, we had an increase in glucose. All right, the second hour, from 3 to 4, look here, all right, the second hour, there was a decrease in the amount of glucose. There we go, we were up here at 1.2, it went down almost to just below 1. Okay, it's actually almost, yeah, we'd say 0.9. So we add our average. Now, you've explained the pattern of the graph, but tell me why. All right, and the reason is that's where you're going to put it over here. So I said, okay, me and this, all these things, okay. All right, all the patterns. This increased, okay, that decreased. Give me the time frame. All right, if you don't put the time frame in, you don't get the marks. Second thing that we need to put in over here, why? So we know that we ate, that's why it went up, because after digestion, carbohydrates, what is everything broken down to? Glucose, all right? Then if those levels stay high, we know it's bad, so we need to get them down, all right? And what kicked in? Insulin, all right? Insulin kicked in and converted glycogen to glucose. Do we have any questions yet?
Yes, we do.
Oh, we do.
Motepa Nathaniel is asking, what happens to adrenaline when a person sees a snake?
Ah, all right. Who's it? that we-
Motepane Daniel.
Okay, Motakani Daniel. Daniel, that was one of the past questions on a question
Really?
paper. Yeah, there's a picture of a little boy, and he's got eyes like this, all right, and then the snake's in front of him. Okay, when it comes to that question, all right, let's start with adrenaline. Okay, go to the functions of adrenaline. Remember, it is your fight. You stay and you fight, all right, which you're going to need strength for, all right, or it's your flight. You need to run away as fast as you can. Okay, so if you are in an emergency situation, what do you want your body to do? Okay, first of all, you want a lot of energy, don't you? All right, you want to be quite strong. So, goes with this. Okay, how do you get strong? You need glucose. All right. So how are you going to get glucose? What's going to happen? Aha, in your pancreas, your liver, all right, your glucose. How do we get it? Glucagon. All right, so in your muscles, in your liver, all right, what is happening? All that nice stored glucose, all of a sudden we take it and we convert that. Also, that stored glycogen. See, I'm even getting confused with the glass. Right, the glycogen, what do? We convert it to glucose. Glucose goes to our muscles, all right? If we've got glucose in our muscles, what can we do? Lots of energy. We can run very far, all right, or we can stand and fight. What else do you need to be able to do, all right? Your heart needs to beat faster, okay? Why does your heart need to beat faster? I'm going to put a heart there and an increase. Your heart needs to beat faster because this glucose is needed for cellular respiration. All right, and in order for you to do stuff, what do you need? You need oxygen. All right, so now we need oxygen. The heart needs to beat that oxygen throughout the body. All right, so it's going to go fast, 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 fast. But I also need to get into the oxygen. So what happens to me? What happens to my breathing? All right, my breathing. I breathe fast and I breathe deep. Okay, fast and deep. Like that, almost like hyperventilating. Oxygen's coming in, right? The heart is pumping it, the muscles are doing the work. What happens to the pupils? They dilate, right? You go into a bit of shock. You're trying to absorb everything. The last thing your body worries about is digestion, those kind of things, all right? So it's not saying to you, oh, let's digest this hamburger that you had for lunch over here. It's not even thinking about that, all right? Or the blood from that's why you sometimes, unfortunately with us white people, it's a bit more easier to see us go pale. But you actually lose, all right, the blood from your face. You go pale, right, and you become sweaty because your body, the paleness, remember we talked about just now, we don't need to have all the blood rushing to our head. Where do we need it? We need it in our muscles, and we need it up here in our brain thinking, and here by our heart and our lungs, right? So the blood vessels constrict, and everything gets shunted to the areas that we need. I hope that answers your question.
Thank you. And then Shikasha's asking, could you please explain how insulin is made from bacteria?
All right. Okay, one of the things when we look at insulin, all right, insulin also connects with our biotechnology section. Okay, now one of the questions there is how, all right, how do we get bacteria to make insulin? And that's going to be in your paper 1 when you get to the DNA and the RNA. Okay, what happens is the following. I think let me just quickly rub out a section over here. Oh no, now I'm going to, that is the eraser, am I right?
Yes.
I think there we go. Oh, I must just tap it. If I tap it, you know what, I'm not going to tap it over there.
Extend the page.
Can I extend the page?
Can you?
No, there we go.
Yeah.
All right, well done, Noonie. All right, this machine and I are mortal enemies. Okay, biotechnology. Bacteria, especially E. coli bacteria, reproduces exceptionally quickly. All right. Now remember, in the good old days, they used to use insulin from pigs, all right, and that whole host of problems, religious reasons, also it wasn't compatible with humans. So what do we have now is a process of recombinant DNA. Okay, so here we have, I'm going to draw the process. Here we have a bacteria cell, all right, And what they usually use is a virus, all right? But it's been neutralized. I'm going to draw it over here. Now, if you know how you did in grade 11, how a virus works, what they do is they take, here's human DNA, all right? They take the information from the human DNA and they cut off that piece, all right? And what they do is they insert it into the virus. It's like a plasmid, all right? They cut it and they call it a plasmid, and they put it into this virus. Now, you know the function of a virus, right, is to change its host DNA. So now this virus is infected, if I might use that word, with human, just a little portion of human DNA that makes insulin, okay? So when the virus is inserted into the bacteria, we know the bacteria has its own, all right, nucleoid of DNA. All right, what it does is this little bit of information combines, recombinant combines with the bacteria's DNA. And the bacteria carries on doing its thing, but now it's a little extra piece of information is being added to it, and now all of a sudden, that bacteria makes human insulin and it secretes it, okay? And they harvest it. Now, because bacteria, right, it multiplies at such an enormous rate, they can harvest as much human insulin as possible, and humans aren't allergic to human insulin. So it's much more compatible.
All right. Thank you. You need to take a break. I'll ask you the
a
other
break.
question
All
after
right.
the break. Thank you so much, Mindsetters, for those questions. Nendlandla, I see your question and we'll answer it after the break. Mindsetters, let's take a short break and we'll see you straight after this. Welcome back, Mindsetters. From the very short break, I see a lot of you guys have questions. We will ask Cheryl when she's done with the section that she's doing now. And Snendlandla, I will answer your question later on as well. Okay. Mindsetters, I see most of you guys are putting your numbers on the post. Don't comment on the post. What you need to do is tag yourself with your favorite number or share the post on your Facebook page and then write down your favorite number so that you can stand a chance to go into the lucky draw to win that very special Learn Extra hamper. So you need to tag or share the post with your favorite number, and then when you stop the clock, it will be over for today's lesson, and then you will watch the following lesson to see if you've won that Learn Extra hamper. Okay, guys? So Cheryl, back to you.
Thank you. I feel so old when she starts to say tag and everything, and I'm like, I have no idea.
New
what
lingo.
I know it's a new lingo. This biology is my lingo. Welcome back, grade 12s. I hope you have so many questions, and I wish that we could answer it all, but Looney is going to answer a few that she can. I've given her a few answers that she needs to, but we're going to carry on, all right, with our section on plant and animal responses. Okay, the next question, all right, if you've looked so long, you should be expecting a question on the eye and a question on the ear, all right? If I have a look at this one, the question that we looked at the ear now, all right, is actually quite a nice, basic, simple question, and it's definitely a question where you should be able to get full marks. Guys, when it comes to your diagrams, if you noticed, almost every single question that we've done is related to a diagram. If you do not have the pictures pasted all over your room, you are really going to battle, okay? So what I suggest, you don't have to be a mastermind, know the picture. Those who I teach know, I'm a great fan of annotated notes, all right? What does that mean? If I had to use here, if you're not an artist, it doesn't matter, you can trace it. But you should have a picture of the ear, even one if you photocopied it, all right? But it's much easier, if you've drawn it, you have a picture in your head. So you take a piece of paper, you draw the ear, all right? Then the first thing that you do is you label it, all right? Use the things in your own handwriting. If you have written it, the chances are that you are much more likely to remember it than if you just look at your textbook and whatever. All right? So you've got a picture in front of you, you've got the ear. You draw it, then you write all the labels all around it. So if I were to do this one, I would write pinna. All right? I would write auditory canal. All right. Eardrum, tympanic membrane. Eustachian tube. Okay, those are the ossicles, the hammer, the anvil, the stirrup. All right, some of you might be taught the stapes, the incus, and the malleus. That is fine. All right, semicircular canals. All right, we know that's the cochlea. Okay, that outer ear, middle ear, inner ear. Okay, so first of all, one, I know the labels, and I guarantee you, if I look here, look at first question for 2 marks, identify part. Those are the easiest of the questions, if you are able to know the labels of the diagrams. Okay, those are given. It's like in maths when you know the theorems. You might not be able to solve that geometry problem. I know I never could, right? You're laughing, Looney. Hey? I never could do geometry. Okay, but at least you have some of the answer you are able to do. All right, and that is pure studying. Then what you do, you can use different colors, whatever. All right, you take each of your labels and you put a function next to it. All right, so here you say collect sound waves. All right, so for each of them, you take your textbook, I'm sure it's got it in, all right, and you put a line, and for each of your labels, you give a function to. All right, here, semicircular canals, you should be talking about balance. All right, cochlea, already you should thinking about hearing. All right, eustachian tube, what does it do? Controls the pressure in the ear. All right, and we know that the ear, all right, is a mechanoreceptor. All right, what does that mean? That all the parts from start, going through, all the parts must be able to move. All right, why do I say that? Because how does the ear work? Vibrations. And what is vibrations? Movement. It is movement. Okay, so unlike the eye, if I shine a torch into your ear, nothing's going to happen because the nerve cells are not sensitive to light. They are sensitive to vibrations. So we need to get vibrations going in because they start the whole movement going, the whole domino effect. Remember, when we're looking at the eye, we want light to come through. Okay, so let's start over here. So for each of these, right, you should have a picture of the brain, different functions of your brain. All right, the cerebrum, the cerebellum, the medulla oblongata, the hypothalamus with the pituitary gland. You should have a diagram of your reflex action, like number labeled 1 through to 5, like I just showed you. There should be a diagram of the eye. There should be a diagram of the ear, okay, that you have drawn. Each of them labeled nicely, all right, and each of the labels, there should be a function supplied to that. If you know that, all right, you really are standing in good stead to do well. And sometimes if you battle with the more interpretive question, stick to what you know. Okay, so identify number B, all right? Number B, there we go, because I've labeled it and I've put it on my ceiling. I see it when I go to bed tonight. I've put it on my bathroom mirror. I see it when I brush my teeth in the morning. All right, so number B, semicircular canals. All right, number C, there we go. I know my structure of the ear because that's the one thing I've done. All right, eustachian tube. All right, 1 mark for each of those. What does structure A consist of? Structure A, all right, are my ossicles. And what are they? The smallest bones in the body. All right, my hammer, my anvil, and my stirrup, my incus, my stapes, and my malleus, they are the 3 smallest bones in the body. Okay, now question number 3. This is where a little bit of interpretation is needed. Okay, now they ask you, explain what would happen if structure D had a hole in it. All right, structure number D is my eardrum or my tympanic membrane. Try and be as biological as possible. My tympanic membrane, all right, which is my eardrum. My whole, how do I hear? Vibrations, sounds, right? Sounds. So what happens over here, when the sound comes through here, what does it knock on over here? It causes vibrations to occur so that impulse can be sent to the rest of the ear. So now, if that has got a hole in it, what will happen? The vibrations, okay, won't be passed on. Keep it nice and simple. All right? You need it to move. And you also need it to amplify, to make the sound louder. All right? So if your tympanic membrane, your eardrum, has a hole in it, all right, the sound waves won't be able to pass through. Vibrations, sound waves, right, won't be able to be passed on. 2 marks. What do you need for your 3rd mark? So, give me a conclusion. They asked you, explain what would happen. All right? So I've said, if there's no hole, well, if there is a hole, the sound waves come and they can't hit against something solid. So if they can't hit against something solid, they can't pass the message on. All right? So if they cannot pass the message on, what is the consequence of this? Deafness, because you need to get it through to the end. All right? Or hearing impeded. What do we mean by hearing impeded? All right? We're saying that you cannot hear properly. Right? What do we need? The vibrations must go. Let's go back to our diagram of the ear. This is where I hear. Okay? The cochlea. Can you remember it? All right? With all its little hair cells. Okay? So we need vibration to go through. If it stops here, nothing's happening over there. All right? And if nothing's happening, then I cannot hear, or the sound is very, very dim, and I can't hear properly. Hearing impeded, cannot hear properly. Do we have any questions?
Yes, we do. Okay, let me just.
It always feels like the hot seat when we answer. And keep praying.
Okay, guys is asking, does. Oh, and then it goes away. Okay, here's a question. What if I wrote ossicles instead of bones? Would it still be right?
No, ossicles better.
Okay.
All right, guys, that's what they're saying. No. Have a look here quickly. All right. Have a look at what does structure A consist of, not what it is called.
Oh, okay.
All right. So be careful that, right, if I was looking at the memo, they were very strict about bones, which means they obviously didn't accept ossicles. Right. If the question was, what are the 3 of them called together, but they ask you, look here, what does the structure A consist of?
Okay.
All right. And that it means bone.
All right.
All
and
right.
then Lisola Mankongi is asking, if a person was involved in a car accident and when a prick test is done on the fingers, they feel the pain of the prick, but when told to move the hand, the person can't. Which part is affected, the brain or motor neuron?
Motor neuron.
Okay. And then Minoli Nigel is asking, can you please ask Cheryl to tell us how much detail we need to know about the inner ear?
About the inner ear. Sure, when it comes to the detail of the inner ear, I find that if you go through, when I say to you go through past papers, it's always, you'll get an idea of the depth in which they need to do it. I, for one, would be able to label it, all right? So the diagram of the inner ear, right, you know when it comes to the cochlea, they've got the 3 compartments there, and they've got the tectoral embryo and the organ of Corti there. I would be able to label it, but when it comes to the ear, they don't always, they might ask you to explain hearing rather than actually all the details. So when it comes to the inner ear, be able to know, all right, where the vibrations began, all right, the endolymph, then the perilymph, all right, the tectoral. So have a flow diagram ready. Get a flow diagram, so eardrum, ossicles, round oval window, all right, endolymph, perilymph, tectoral membrane, organ of Corti, all right.
All right.
Okay, that's a very tricky one. They don't, you must know it, but they don't always tend to go into it in too much detail. Do know sometimes the balance, all right, go into the balance a little bit more because it links in with the brain.
Okay. And then Snentland Latemban is asking, what is the difference between hypothermia and hyperthermia?
Hyper and?
Hyper. It's hypo with an O first and then with an A.
Okay.
Hyper
it
and
should be hypothermia and hyperthermia.
Hyper, okay.
All right.
Mistake
so
there.
hypothermia, talking about too hot. Hypothermia, talking about too cold.
Okay.
All right.
And then, Spusis Tetro is saying this is out of topic, but he's asking how does cloning differ from artificial selection?
Oh, okay. All right. When you clone something, all right, cloning, you have an organism already. Okay, so cloning actually involves changing, you actually fiddle a bit with the DNA. Let's start with cloning. Okay, when you have cloning, I'm going to, let's see if we can extend the page here. There we go. All right. When you have a cloning, you have 2 different cells. All right. One of them is a body cell. Now, the reason why we use a body cell is because it's 2N. Okay. So a body cell has already got, so say you want to clone me. All right. My husband would just die. He'd probably be ragged to death. All right. So if I wanted to clone myself, if I take one of my cells, I know exactly what it's going to look like. Voila. Here she is, right over here. Okay. No surprises because all my DNA is already there. It's a blueprint. Okay. Now, what do? Here is another egg cell. I take out all the DNA. All right. I don't want any because I'm going to turn into a monster. So it's a blank. Okay. And when these 2 join, they shot of electricity, and it simulates fertilization, and you already know what genetic you have got. Artificial selection, is that what they?
Yes.
Okay.
Yes.
When you artificial selection, you don't interfere with the genes at all. All right. You choose an individual that is best suited for what you're looking for. Okay. Let's use the good old version. You are a dairy farmer. You want cows that produce milk. Okay? So you've got all of these cows, and you see one cow over there. She produces a lot of milk. All right? This little cow over here, although it looks nice and big, she hardly produces any milk. So obviously, this one that's nice and big with lots of milk, she has got the genes. She's genetically able to make a lot of milk. Right? So I take her, and what do? I breed with her. So what gene have I got? I've got the nice big milk-bearing gene. And what do I breed? Because I'm hoping that each time her milk-bearing gene will be passed on, obviously, to her offspring. That is artificial. All right? If you want a dog to protect your house, you're not going to have a little chihuahua. All right? What do you need it for? You want something big. So you breed with something big. Okay, so you choose the best traits. If you want to grow a lot of grapes, you have a vine that has lots of grapes, because the chances are when you plant it, it will grow a lot of fruit again.
All right.
Okay.
Let's ask the last question before we go to a break. Guys, Atlalele is asking, is insulin produced in adult or newborn? I'm confused now.
Insulin is produced from the beginning, all right? Insulin is produced from the beginning in the pancreas, right? There are alpha and there are beta cells, and but it becomes more prevalent when they become children and they start having a diet rich in carbohydrates. Remember in the beginning, not so much carbohydrates, but lots of proteins.
All right, I think that's it.
Is that it?
Yes.
All right.
Should we take a
We're
break?
taking another break.
All right. Mindsetters, we are going to take a break, but one thing I want to say, how easy does Cheryl make life science seem? You ask her a question and she just answers, it just flows out of her mouth. That is very fascinating. Awesome teacher we have right there. Anyway, mindsetters, we are going to take a very short break, so do stay tuned. We will be back straight after this. Welcome back, Mindsetters. You know, sometimes I always say, I wish you guys would see what happens here during the break. You would laugh so loud. But anyways, remember to enter, guys. We haven't stopped the clock, so please just tag and share, and you will be entered into the lucky draw. Remember, 0 to 9 must be your favorite number. You tag yourself, you tell us your number, you share, you write your number, and then you'll be entered into the lucky draw. If we stop the clock and that's your number. Our clock is our Facebook likes. Remember the last digit of our likes. So say maybe it's 54,116. 6 will be the last number. So that's the clock where it will stop. So I hope you guys understand. I see most of you have been posting it on our wall and on the post. Don't do that, guys. Tag or share, okay? And I hope you guys understand the lesson. And thank you so much for tuning in to everyone who's on the page and commenting and talking to me. It's nice to have people talking to me and Cheryl, to our awesome Cheryl. So I'll take it straight back to her.
Thank you. Sometimes when you see what goes on behind the scenes, we have an awesome team here of James and Abram. They're just so funny. They make us laugh. All right, guys. When we look carrying on, all right, you know we've covered the ear. Okay, the next question is going to be a question on the eye. When it gets to the eye, what I would concentrate on again, first thing please, you have your diagram of the eye, you've labeled it, sclera, choroid, retina, all right, iris, pupil, ciliary body, lens, cornea, aqueous humor, vitreous humor. You must know, and then go functions of each. Okay, but the question on the eye here is taking on a bit of an investigative. Remember, a science investigation is also part of the skills that you need to use. Okay. I'm going to go through this too quickly, but I do want to spend a little bit of time on the essay for you, because I know for most of you, all right, the essay, it's only 20 marks out of the paper, but I think it's the one that you stress about the most. Okay, so let's, Karen, this question made me laugh, sorry, because the person's name's Topsy. I know, me and Luvi, I was trying to ask, maybe it was like a secret character that I didn't know anything about, but apparently Topsy. Okay, so now we're looking at determine the effect, all right, of distance on the curvature of the lens. Here we have the lens, all right, of our eye, and now you need to start thinking what changes curvature. My eye can be elongated, all right, or me in this board, or it can be, all right, much fatter. Okay, now we know that it changes. How does it, when does it change? Start thinking, all right, accommodation. All right, accommodation. Remember, if anything comes 6 meters and closer, my eye needs to accommodate. My lens needs to become more convex, right, so that I can put that image onto my retina. Okay, but this is a bit of an investigation. All right, what they found, old Topsy over here, all right. Okay, let's have a look. She was in a well-lit room. They covered her one eye with an eye patch, all right. There was a pencil held in front of her uncovered eye for 10 seconds. That's important. Remember, you are doing a science experiment and you want your variables to be constant. She focused on the pencil until a clear image could be seen. And what did they do? They measured the curvature, all right, of her lens. The pencil was then moved to different distances, right, from the eye, and the curvature of the lens was measured. So basically, if this was my pencil, all right, they made her cover the eye, right, she had to focus on it for a certain amount of time, and at each time, they moved it further away, okay? Look, what we're looking at, accommodation, distance, all right, what happens to the lens when something comes closer and when something goes far away. Now they give you the results. All right, what do you notice? Now have a look. right? They could have asked you to put this into a graph, right? But it's giving you the answers here. They're telling you exactly what happened. Okay, so at 10 centimeters, the curvature was 4 millimeters. So when we talk about that, we're saying that it's fat, all right? At 200 centimeters, the curvature was less. Closer, fatter, further away, all right, the less the curvature. So now, let's answer the questions. And this is the one that you guys always get confused about. Okay, dependent variable are the results. All right, the independent variable, I always remember I chose. What of the characteristics could I control? All right, now remember, here we go. When you have a table, the first column is always, or should be, if they're correct, your independent variable. Didn't I control it? What did I decide? Okay, 10 centimeters, 20 centimeters, 30 centimeters, 40 centimeters. That was my decision, or whoever was running that was the controlled. I chose it. Okay, what could I not control? My curvature of the lens. I didn't know what my lens would do if I held it there or if I held it over there. Those are the results, all right? So your second column should always have the dependent variable, all right? Independent, dependent. So here we go. Which is the dependent variable? Let's go. There's the heading of my, all right, curvature of my lens, all right? Which is the independent variable? What did I decide on? Distance. Okay? I decided here, 10, 20, 30, 40, but the curvature, that is what I wanted to test. That is what I was looking for, right? Does the curvature, does the distance have an effect on the curvature? Okay, now it is an experiment. State two factors that must be kept constant. Okay, what must we do? First of all, we must use the same eye. Okay, right, that sounds really stupid, but if I'm going to do this, and then I'm going to do this for one, it's totally different, all right? Because I know for me, this one over here, my right eye is much stronger. If I close, this is much more difficult. Okay, second of all, what else? Hold the pencil for the same amount of time, 10 seconds. Finished. Then move 10 seconds. So the eye has exactly the same amount of time to adjust. Okay, so time. All right, what else? What else must we use? The same pencil. I know that sounds a bit doof, right? But it's the same thing. It's the same size, the same shape, everything. All right, so we keep everything, all right? We want to make sure our results are, all right, credible. They are valid. Same pencil. All right, have a look at this one. Oh, there we go. Sat in a well-lit room. Same amount of light. Right? Our eye will react differently if it's light or dark. Right? So we want each time I move my pencil, everything, the conditions must be the same. Same pencil, same lighting, all right? I hold it at the same amount of time. Now, remember, whenever it is in capital letters, what does that mean? It means you must only write 2. They only mark 2. If you write more, all right, and your first 2 are wrong, and your rest are right, sorry for you. They only mark the first 2. All right. Explain why the factors named must be kept constant. Always, it's an experiment. Why do we keep certain factors constant? To make our experiments results valid. All right, for validity. All right, if I have kept everything constant, then the chances are that my results are going to be valid. All right? They're not going to be, all right, I just put anything together and the results could be anything. Okay. Now, the next question is actually for 4 marks. Describe the relationship between the distance of the pencil from the eye and the curvature of the lens. What they're trying to tell you here, look, what is the relationship? When the pencil is closer, the curvature is more. So you write that, all right? Or you can write the opposite. The further away the pencil moves, all right? The further away the pencil moves, the curvature gets less. All right, there we go. The further away the pencil moves, your 2 marks. There's an until. Have a look here. What do you notice? What happens at 150 and 200? They stay the same. Explain it. Describe the relationship. Until 150 centimeters, the lens no longer changes curvature. All right. Okay. Last question. Name 2 structures, 2 capital letters. You can't list every single structure in the eye and hope that they're going to find the right answer in there somewhere. Your first 2 answers. All right? And if we look at the lens, what holds them in place? All right? And what pulls them? The ciliary muscle. Okay, there, the muscle. And what hangs on the lens? The suspensory ligaments. Okay, questions?
Yes. Piwem Bangan is asking, what happens when a person has a headache?
Okay, James is whispering in my ear that I have to answer after the ad break.
In your ear.
And you don't, when James talks, you listen.
Yes. All right. My series, you heard it. If you don't know by now, we have people talking in our ear.
Yeah.
So when you see us pause sometimes and listen, you must know we're multitasking. It reminds you to we are going to take a very short break, so don't go anywhere. Welcome back, Mindsetters. We are in the final leg of this life sciences race. I also did this with the math, so I hope you guys are enjoying it. We're in this last leg, guys, so I hope you guys are enjoying the show. Make sure you tag yourself and share because we are going to stop the clock sometime before the show ends. So, Cheryl, over to you.
Hi. Yo, there are so many questions that you guys have. I wish I could just sit down and answer them all, but I do think there are people who look at the questions and who do answer you. All right. If not, I'm going to speak to John and we'll come up with a plan. We need to get to our last section, which is the essay section. And as I said, I think that's the one section where you guys are a little bit, you get a little bit scared about. All right. I know I used to write biology before most of you were born. Our essay was for 75 marks, and it used to kill me. Okay. James says we have to do the headache question. All right.
All right.
Okay. What is a headache? A headache, quite simply, remember you've got pain receptors, all right, that all over your body. You've got the pain receptors. And quite simply, what a headache is, right, is when those pain receptors are stimulated. Right, so in your head, it could be things that you've eaten. It could be muscles that are on a nerve, right, and what happens? Those little nerves, they are triggered, and that you perceive that as a headache, all right? That's why you have a headache tablet, because remember, when you have a nerve, when you did the nervous system, what must happen, all right, one nerve joins with another one, there's a little bit of a gap, and that chemical in the middle passes that, all right, impulse on. So when you have a headache tablet, it stops that chemical, right? It deadens that chemical for working. So the impulse can't travel, and then you can't feel the perception of pain.
Thank
Okay, James, is that fine now? Can we go on? It's like the voice in our head. All right, guys, this again, when it comes to the hormones, this is a very important question. It's the negative feedback mechanism all right, involving thyroid stimulating hormone and thyroxine. Now, if you have a look at this question, it is twofold, okay? Number one, you must describe the negative feedback mechanism. That's it. You'll see over here. Let me go. John has put it perfectly. There we go. We've got such amazing people here. All right. When we look at the question, describe the negative feedback mechanism involving TSH and thyroxine, and describe the consequences if this mechanism does not function well. Okay. Your essay is out of 17. All right. It's always out of 17. 3 marks is for how you wrote it, okay? But and it's 17 facts, right? We want 17 facts. So you'll see, comes up with the essay, there's a lot of things that you can write, but you just must make sense. Let's answer the first question, all right? The first one, this is so nice, John put all this up for me. The first thing which we must know, all right?
Cheryl?
Yes.
Could you please just, before you carry on, can
Yep.
you just quickly stop the clock so that we have our entries for this show?
Okay, so
If
do
you
I
could
go
just
here?
Go to, and
And then what
refresh
do I
the
do?
page quickly.
Okay, refresh
Yes.
at the top.
Yes.
Okay.
Okay, so it's on 8. So guys, it stopped. We've stopped on 8. That's it. 8 is the final number, so we're taking that. So thank you. You can carry on, Cheryl.
Okay.
Thank
Okay,
you.
I think they do this on purpose. All right. Okay, let's go. TSH, I'm going to draw a picture here. Remember, here's the pituitary gland. Here's your thyroid gland. Okay? So we know that the pituitary gland is sensitive. The model answer is here. John is so clever to do this for us. The pituitary answer is sensitive to the concentration of the hormone thyroxine. That's it, okay? Now describe the whole process. Now listen very, very carefully. At the bottom of your question, it says there, no flow diagrams. You must, even if you do it in point form, all right, put it down point by point. That's fine. Do not draw any kind of diagram. Okay, so let's start. The first thing, all right, what happens? Let's start off when the level is low, all right? What happens when our level is low? So we start. Pituitary gland secretes thyroid stimulating hormone. Where does it go? To the thyroid gland. What does the thyroid gland do? It stimulates thyroxine. Now what happens? The thyroxine levels are high in the blood. Look at that. Look how many marks that is. Secrete more TSH. TSH stimulates the thyroid gland, secretes more thyroxine, increasing the level, and now it's normal. We haven't even only started, and look how much we've got. All you're doing is you're taking that diagram, pituitary, TSH to thyroid. Thyroid produces thyroxine, thyroxine increases, then what we have, normal blood level. Right? Now, when the levels are too high, we must have another, remember, homeostasis. Now, what happens when our levels are low? Right? When our levels are too high. Right? I need to get them down. So what do? I shut off the switch at my pituitary gland. So, pituitary gland not stimulated. If I've got no pituitary gland, here's my pituitary gland, what am I not going to have? No TSH, all right? No thyroid stimulating hormone. No thyroid stimulating hormone, the thyroid cannot produce thyroxine. So what happens? My level goes down, okay? Remember, level high, TSH, stop. Level low, TSH, start. So there we go. Look what we've got. It's not stimulated, all right, less stimulation to secrete thyroxine, decreasing. Okay, but that's only half of our essay. All right, what was the other half? Let's have a look. Describe the consequences if this mechanism does not work. All right, and when you did hormones, what did you look at? What happens if there was too much, and what happens if there was too little? So we start. Here we go. what? Not working. The concentration of thyroxine will not be regulated. If it's not working, my thyroid is not producing thyroxine. Okay? Or it's producing too much. So let's look at if it produces too much. Hyperthyroidism. Okay, so there you're telling me it produces too much. What else are you telling me? All right? Increase. Give me the symptoms. Now, too much. What does the thyroid control? My metabolism. So what happens to my metabolism? It increases. With this high increase, remember now you're looking like, like something that's gone haywire, right? What do? You're hyperactive, right? Because you're hyperactive, you're using all your energy, so you start to get thin. You have protruding eyes, all right? And because you're always working, you also suffer from insomnia. You don't sleep. So because you don't sleep, on the other hand, you're tired, all right? Your heart rate is beating all the time. Remember, it's like you're on super alert, okay? So now, which is not very good for you because it also can have a problem with your heart. So there we go. What else? So if that's too much, if I've got too little, hypo, look, hypo too little, hypothyroidism, and it decreases. Remember your thyroid gland, we want metabolism. Now it's not working. Now what do? All right, I'm slow, I'm sluggish, I'm tired, I gain weight, my heart rate decreases, all right, and slow growth of hair and nails. So in babies, it also can cause cretinism where they are mentally retarded. Are we got more questions?
Yes. Snantland is asking, can you please explain pupillary mechanism?
Okay. One question, James says.
Yes.
Okay, pupillary mechanism, all right, remember, is how your pupil constricts or dilates. Your pupil is a black hole. It can't do anything. What is around your pupil? Your iris. And your iris is made up of muscles, all right? Circular muscles and longitudinal muscles. So when you go out into the light, too bright, what happens? Circular muscles contract and your pupil becomes smaller. When you go into the dark, you want to get more light. So this pupil needs to widen, longitudinal muscles contract, and your pupil dilates.
All right.
It's the iris, not the pupil.
Yes.
Okay.
Thank you so much, Cheryl.
Pleasure.
For being so awesome today.
Oh.
Mindsetters, thank you so much for tuning in and thank you for entering the competition. We will send all those names into our lucky draw that's coming up next in the next show. So check this out and you might win yourself a Learn Extra hamper. Thank you for tuning in. Everything of the best for your final exams. Person in my ear says, "Iskhathi siphelile," so we are out, guys.